Let's
Yes,
start.
sir.
Good morning, everyone. Again, today we'll be unlocking all the difficulties, especially with the monohybrid, dihybrid, and of course the codon sequence. Right? So these are important details, and these will come out in your second mastery test, and of course in your major exams. Okay. Now, for let's go back uh, to the monohybrid. Okay. Again, monohybrid, these are the basics. I'll be sharing my screen to you. We'll be using a Word document in order for us to evaluate. Okay. Just, I will just put this, I will be discussing that one later. Okay, let's go back to monohybrid. All right. In your master tests, I already given you uh, I already given you the constant variables or the variables itself, right? In your major exam, like your um, quarter exam, I'll be giving you a problem and you'll be solving that one through the use of monohybrid and dihybrid cross matching. Okay? So these are the basics only, right? Now, confusions with the monohybrid. Let's start first with the monohybrid. Like for example, uh, let's take this allele. Mm, hemophilic. Okay, hemophilia is a case where a person usually have nosebleeds. Okay, that's hemophilia. Okay, now hemophilia, there are some cases that hemophilia uh, becomes dominant over with the other genes. Okay, meaning to say that it came from a homozygous hemophilic parents. Let's have this one example. So this is hemophilic. Can you see my screen?
Yes, sir. Yes.
Hemophilic, and the other one is non-hemophilic. Let's just use the simplest form. Maglibog na sa niya mo ani. Let's have, what else? What the color of the eyes? No.
Sir, hemophilics are the one who are more exposed in nosebleeds.
Yes. That's the persons usually have nosebleeds. Okay? Those are genetic. Do you have hemophilia?
No, sir, I'm just asking.
Alright. So, let's have na lang, uh,
So, sir, there are different names in each genes.
of
Like
course.
even the like this.
Of course. Yes, yes. Yes. Let's have, oh, the color of the eyes in Drosophila melanogasters. Drosophila melanogasters, these are fruit flies. Kana bitaw makita ninyo sa banana, okay? The fruit flies. We usually use that one for genetics because their their eye color, let's have red. R is for red, and this is for the Drosophila melanogasters eye. Kana bitong sa inyong fruit fly, when you see a banana, no? When you leave your banana for how many hours, there are some flies there. Kanang gagmay na fly ba? And those are
Yes, sir.
Drosophila melanogasters. Okay? Those are fruit flies. Small letter R or white. Okay? This eyes ha. Okay. We will now cross match a Drosophila, which is a heterozygous red, and to a Drosophila, which is a homozygous red. What could be the result? Okay. possible offspring. Okay, let's have this Punnett square. Again, please let me know if I'm too fast. Okay, we'll be taking this little by little. This is a review for you. We already discussed this one. Let's have this one. I'll be inserting some tables. Okay, male, we have a heterozygous red, and the female is homozygous red. The results are,
Excuse me, sir.
yes, yes, Margie.
If the kwan, tawag ni- if the 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 heterozygous red, sir, it's in the left. It will be always on the top
Oh wait,
using the square.
you
When
said
Sir,
using
what?
the
what's
square,
heterozygous and homozygous again, sir?
Let's have this one ha to have a uniform idea. Okay, in that area, Margie, those are the male, no, the male. Okay, on the top area is the male, on the side area is the female. Okay, let's have a uniform idea. Okay,
how about that part, sir?
what part, Margie?
Ay, the
One.
big R and small RX, 2 R's.
Okay, the big and the small r, diba? This is heterozygous. This is actually the male, right? And the
If
home
it's always on the left, sir, it's the male.
on the top is the male. Take a look at this one. This
Eh,
is the male, and this is the female.
So those are called heterozygous and homozygous, sir.
Okay, di ba we are done with homo means one. When you say homozygous, it means one allele. So, like for example, the female here, this is considered as homozygous because it only contains a gene for red. It doesn't
One.
contain a gene for
Yes.
white. Correct? Okay. Now, the male here is a heterozygous. When you say heterozygous, it actually contains two or more alleles or genes. Like for example, the male here has a red gene or allele, at the same time carrying now the recessive gene which is
Ah,
white,
okay, sir.
heterozygous.
Okay, sir, understand.
Okay. Going back to the discussion, if you're going to cross match a male, uh, Drosophila melanogaster with heterozygous red eyes, and a female Drosophila melanogaster with homozygous red eye, what could be the result? Okay, the results are homozygous red, okay, homozygous red, heterozygous red, hetero because this is uh, an allele for red and an allele for white, so it's a mixture between red and white. Okay, again, homozygous red because it actually forms or contains only red. Okay,
One.
allele.
Yes.
Okay, and the last one is a heterozygous red. Okay, if you're going to look at it in a genotypic ratio, diba? We're talking about genotypes. Genotypic ratio. It is genotypes. Genotypes means
To whom
what's with the genes. Okay, we're talking about genes. Two, Margie, yes. What could what,
homozygous red,
homozygous red? Okay, correct, Margie. is two, Margie?
Two heterozygous red.
Two correct, zygous red. Two heterozygous red. How about if you're going to look at it in the phenotypic ratio or phenotypes? What can you see? What can you see there in the phenotype? What can you see there in the phenotypic ratio? Anybody? Margie?
Two red. Two red.
Two red. This is red. Margie, you listen.
And
Yes, red.
two.
Okay, so let's count the number of red. Okay, one, two, three, four. It's actually four red. Even though it is heterozygous, it is still red, but it is carrying the gene for white. Okay, four red or 100%
For white.
red. In genotypic ratio, two homozygous red and two heterozygous red, or 50% homozygous red is to 50% heterozygous red.
Sir, why is it 100% red? Because the red is dominant than the white?
No, this is red. Okay, right? This is homozygous red, so it's red.
Yes.
Also red, correct? How about this one? Is this red?
Yes.
Okay, is this red?
Yes, sir. Yes.
Okay, so therefore it's 100% red. If you're going to look at it in a phenotypic ratio. Again, what is phenotypes again? When you're talking about phenotypes, you're talking about what you can see or observe or observable traits or behavior. Like for example, the color of your eyes, the color of your skin, what else? The shape of your face, what else? Uh, your hair, it could be curly or straight. Those are phenotypes. Okay. Now, when you're talking about genotypes, you are actually talking about what's inside that gene. Even though you have a straight hair, but you are carrying a gene for curly hair, so you can transfer that gene to your offspring or to the next generation. Okay. Any other question? Nasabtan?
Can you
Sir,
give
can you
another
do another
example, sir?
example, sir?
Another example?
Yes, sir.
Okay. Let's have what else?
With four quants, sir. I'm confused with that.
Asa dapita ka na confused, Margie?
Kanang four ka quants, sir.
Porca red?
Dili, sir. Sa kanang
Punnett Square? Margie?
Sir, yes, sir.
Okay, asa mo naglibog sa Punnett Square? Again, this is the, ako i-color ha. Okay, this is, uh, ato lang siya kuan mo. Ito lang siya i-fill. We'll be shading this one with blue, means paternal or father or male. Okay, I'll be shading this one with red. Red means mother or maternal or female. Okay. Asa man mo na confused David? The mixing? Ano raman na oh? Okay. Kini na R atong gi-transfer here and the R is transferred here. That's why we have homozygous. Red. Another this R is actually transferred here and the R on the father side is transferred here. So we have heterozygous. Okay. Again, another R from the mother is transferred here and another R from the father is transferred here. So you have homozygous red. And again, another R from the mother and another R for the father. So we have heterozygous red. So asa napita na confused?
I typed it in the chat box, sir.
Okay. Ah, sige. We'll be using that one. Okay. This is what we call as what? This is dihybrid. Okay. When we say dihybrid, it's two or more alleles. Okay. Let's have this one. Same example. Okay. A Drosophila melanogaster. I'll just erase this one. Same example, Drosophila melanogaster. Red, white for the eyes and uh, long and short because some of the Drosophila has a long body or a thorax, long thorax, thoracic cavity, and a short thorax. Okay, let's have this one for the color of the eyes and the thoracic cavity. Thoracic cavity which is long and short. Okay, let's now match a Drosophila with heterozygous red eyes with heterozygous long to a Drosophila which is a heterozygous with a heterozygous red eyes and short thorax or thoracic cavity. Okay, let's have this example. Again, we're still in the Drosophila melanogaster, fruit fly. Okay. Now, in crossing dihybrid, you need to cross first this one, the two alleles again. First step is to cross first the two alleles in a monohybrid setting. So let's have this one. We will cross match this one. Take a look at in order for you to get the variables. We're now talking about variables. Okay. Let's take this little by little. Okay. We will cross now two alleles of the male parent. Okay. Cross. The results are okay. These are the variables for your male. Okay. So I'll be writing this over. Kung na siyang isuwat dire. Okay, para masabtan. RL. Okay. LR. LR. So we are done now with the variables for the male. This is now the variables for the male. Okay. How about the female? So let's erase this one. Replace with the female. Okay. R and um, a short. Okay. Let's have this one.
This is how we solve answer with the hybrid.
Yes, this is
First,
the
we have
male.
to solve the male's con.
Male and female. You need to get the variables first because you cannot cross without getting the variables. You are now getting the variables.
So sir, you have to do it by male and female.
Yes, yes. There's a need.
Like the more separation separates like ikuan ba?
I-separate sa ninyo o cross ang male itself and the female itself. Like what we are doing now. Okay, we are now separating or extracting rather the variables. Okay, this is now variables. So our variables for female we have RL, LR, IR, and IIR.
Excuse me, sir. The other one is RI, but why is the other
Oh,
one is
LR,
IR?
that's LR. LR. RL, the small letter L, LR, small letter L, with this is what we call as the gene for a red and white, and red and white, then red and white. Okay? So para di maglibog, balik ko na to. I will erase this one. Okay? You look at the pattern. Okay? I have seen some of you are really confused. Okay? Crossing match. We need to consider the primary gene and the secondary gene. So, we will have the primary gene sa red. Okay? And the secondary gene is the thorax or thoracic cavity, which is long and short. Okay? We will cross again the male. So, the gene for
R.
uh, the eyes and the gene for the thoracic cavity. Okay? This is L, small letter L. Okay? Because we have heterozygous red and heterozygous long. Okay? Take note, we will first write the first gene or the primary gene which is red, ba? The color of the eyes. Okay. Let's write the first gene to the second gene. Again, the first gene to the second gene. The first gene to the second gene. First gene to the second gene.
Okay, okay, okay.
This one. Okay, if you're going to scramble it up, I think you'll find it hard. But in this way, it is understandable. Yes, Margie.
Sir, the
Nasabtan
it's
ra?
always the male that's dominant, right, sir? Because the small R answer is first before the
Mm-hmm.
capital L.
Mm-hmm.
So sir, we are talking about the male.
We are still talking about the male. And we are now
Okay, sir.
prioritizing what is the first allele. Okay, the first allele is the color of the eyes. And the second allele is the thoracic cavity. Okay?
Okay, sir. That's why I was confused because
Mm,
the last we were talking about the female. I thought we were still talking about the male.
okay. So let's extract now the the factors or the genes for male. Okay, we have RL. Remember, sure, I thought dominant ang L, 'di ba? Capital. No. Take a look at the firsts. Again, we will start at the firsts or the primary gene, which is your red,
Male.
the eyes, 'di ba?
Yes, that.
Eyes and thorax or body. Okay? Eyes and body. Okay? Next is the eyes and the body. Same with the female. Okay? You do the same with the female. Okay? Again, we'll erase this one. Female. We have the color of the eyes, which is heterozygous red, and the thoracic cavity or the body. Okay, again, let's start with the eyes. Eyes, body. Eyes, body. Okay, eyes, body, and eyes, body. So, the factors now or the genes for the female are the
R RL.
RL or the
Small
eyes
letter,
and body. Okay,
small letter R
R
and L.
L R L R L. So we are now ready crossing now the hybrid. Okay, it is now ready for the hybrid. Let's remove this one. Let's move now to crossing dihybrid. Okay. In crossing dihybrid, you need to put all the variables. Okay. We will put now all the variables, starting with this is the male, right? Okay, we'll color this one blue. And we'll color this one red. Or shall we say yellow? For the male, we have RL. Okay. RL. 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 Is that correct? Double check.
Yes, sir.
Okay.
Yes, sir.
Let's move now to the mother or the female. Okay. RL. Okay. RL. 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 Double check. Is that correct?
Yes, sir.
Yes, sir.
So we are now already crossing now the two alleles, right? Starting with the male. Okay. The male has in this quarter or in this box or cell, we have RR. So the first is homozygous red with a heterozygous long. Correct?
How about the short, sir? There's Iman.
Oh, yeah, that's why. This one, di ba? This one. It's not IL. This one. Sakto?
The L is it?
Sakto? Okay?
Yes.
Yes.
Again, in this area or in this lesson, we are not prioritizing homozygous or prioritizing dominance and recessiveness. So the capital
Ah,
letter should start, okay, because it's dominant over the
the
small
small.
letters. Okay, we are now focusing on dominance and recessiveness. Okay, let's move now to the second cell. Okay, heterozygous red with a
White.
heterozygous long. Correct? It is still
Yes.
heterozygous. Heterozygous, the first one is homozygous. The second one is heterozygous red. and heterozygous long. How about in the third cell, we have
Hetero
homozygous red
R
with
and heterozygous.
short. Could you still follow me? Naglibog?
Why, sir? Why is it RR?
RR. So where does the RR came from? One R and the second R.
R.
So it's RR or homozygous.
Okay, sir.
Correct? Sige. Please ask questions so that we are actually riding in the same plane. Okay? Please ask questions. I will appreciate it if you ask question. Again, the R from the male and the other R from the female. Okay? The short gene from the male and the short gene from the female. Okay, to make it a homozygous red.
How about the small letter L, sir? From the first.
This one, there's it. And this one. Diba?
No. Go back to the first one, sir, ba?
First
The
one,
RRL.
this one.
Yes, that one.
Okay. This R is from the male. The second R is from the female. The capital letter L is from the male. And the small letter L is from the female. Correct? Sakto?
Yes, sir.
Okay. So let's continue. By the way, if this Zoom meeting will be
So,
let's continue
sir,
to reconnect. Yes.
so nasa tunga-tunga ang ibutang nimo sa kuan between the two the two R and I nasa tunga mo ibutang.
No. Remember the dominance and recessiveness?
Sa tunga.
When you are talking about um, genes, there are some genes that are dominant, there are some genes that are recessive. So, ang first yun mong isuwat if dominant, if genes gani is the dominant. That's why capital maguna. Okay? If all small letters, then all small letters. But if it is
Sir.
recessive or shall we say heterozygous, it's capital letter first. Yes. Another question before we continue.
Sir.
If the Zoom ends, sir, we will come back.
Yes, please come back because we will continue discussing this one. What is
Sir.
your subject, by the way?
Oo na, sir. You're the last subject for
Okay,
this
okay.
morning.
So that's good. Sige. Again, yes, any other question before we continue?
Sir.
Yes.
The male should always be in the top tier.
Again?
Ang male dapat sa babaw. Major.
We should have a uniform sa these are the basics. We will put the male on the top and the side are the females. Okay?
So sir,
Yes.
the next one is R, then L, then the capital letter L, then the small letter L.
Next, this box. Where's the capital letter L? There's no
Yes,
capital
the
letter L there.
capital.
These are small
Oh,
letter L. So meaning to say this is
okay. So capital R, then small R, like that.
okay. This is considered short. Okay, we have heterozygous red eyes with a thorax short. Okay, another box or another cell we have says can you provide me with the answer? Any
The letter
volunteer?
R,
Small letter R.
capital.
Okay, capital letter R and next
Small R.
small
Small
R.
letter R, then big letter R, then big letter L, then small letter L.
Correct. So this is now heterozygous red with a heterozygous long thorax body or thoracic cavity. Okay, next. Next.
Big letter R.
Where's the big letter R there? Take a look at the male and take a look at the female.
So capital letter L first, sir.
Diba? Remember that we should first prioritize the primary gene. What is the